It's really great to be here, to be back here. I was telling a few people this morning that I was actually one of the part of the first batch of people who moved into this building back in 2012. I actually came here even before the building opened, just to, talk to the architects and figure out where to put things as we were going to be setting up the lab. And then I stayed, I worked here until 2016, and I moved now almost 10 years ago, and it's great to be back. So thank you very much for the introduction. I'm going to be talking to you. You notice I'll change my title a little bit. I'm going to be talking to you about a variety of things that we're currently doing in the lab. And the title reflects kind of the unifying thread of the lab, which is studying cognitive resilience and how different factors, environmental factors, can influence cognition. And we mostly study the hippocampus, which is this beautiful structure, one of the most beautiful structures in the brain that is displayed here from the mouse brain. So I probably don't need to tell this audience that environment, disease, and aging can have a major impact on cognition. This is a paper from New England Journal of Medicine from researchers at Einstein from a few years back, just detailing how even leisure activities, whether it's exercise, social interaction, and so forth, can affect the risk of dementia in elderly populations. So we've known for a very long time that environmental factors such as cognitive enrichment, exercise, social interaction, also aging, and also disease and aging can influence cognition. And we are looking at all of these aspects and trying to figure out how they affect spatial memory, and in particular the encoding of space in the hippocampus. And the reason why we're studying spatial memory is for two reasons. One of them, in neurodegenerative disorders, is one of the first aspects of memory that starts going away. But also it's because it's a relatively well understood system in the hippocampus. And the reason why we understand it pretty well is because of the work, among others, of John O'Keefe, who won the Nobel Prize back in, I believe, in 2014, for his discovery of place cells. So in the hippocampus, in a mammalian hippocampus, there are cells that encode the position of animals in space, right? So these will be cells that respond to the mouse when the fire specifically when the mouse is at a particular location. So if you have a mouse going around a track, there will be cells that respond selectively to each location. And you can see it here in 2D. For example, the cell 1 only responds if the mouse is in a cage, only responds in this corner of the cage, and cell 2 responds in this other position, and cell 3 at another position. So there's a certain specificity to the firing of these cells. And we think that this is how spatial memories are formed. And in fact, if you disrupt this tuned firing to place, you disrupt spatial formation of spatial memories. So the method that we use for our experiments, I'm going to be showing you a few vignettes of the work that we do. So I'm going to go a little bit over the methods first. And the method that we use for our experiment is to record the activity of neurons in the hippocampus of mice. And we record this activity by implanting a window implant with a glass bottom. And this is a chronic implant. We remove a little bit of the cortex of the mice in order to do this implantation procedure. But then the mice go back to their cages, and we can keep them and image them for a long time after implantation. And this allows us to image both the CA1 and the dentate gyres, and even CA3 and other parts of the hippocampus. And we can image this day in, day out, and we can express calcium indicators in these cells. And the calcium indicators, their signal will be a proxy for neuronal activity. So we can see what neurons are active. And you can do this during actual behavior, right? So we can attach the mouse to a microscope, to a treadmill on the microscope, and have the mouse run on the treadmill. as we're imaging it. And we can actually do recordings. We can actually do make the mouse engage with spatial tasks, spatial memory tasks as they're doing this. In this case, for example, the mice are running on the treadmill, and you see here they're running for a water reward, a sucrose water reward. In the blue lines are a water reward, and the black ticks here, these are when the mouse is actually licking the spout. So you see that the mouse starts licking the spout just before it arrives to a location of the reward. And here, this is the movement. So you see it runs, it stops at the reward location, then it starts running again it doesn't stop. It knows that it will only get a reward at a particular position, and it keeps going always to the reward location. And through the efforts of many people in the lab, we can now do this not only in mice that are young, but I believe we're maybe the only ones to do this kind of head fixed behavior and image hippocampus in mice that are over 20 months of age. So their age, and they are running on a treadmill, and they're performing this task even despite their relatively advanced age. So, and we actually can find deficits in the learning. So we usually train the animals to lick at a particular location, and we measure how well the animals can learn a new location of reward. So we switch a location of reward, and young mice, very quickly, they adapt. They start licking at the new location and stop licking at the old location. Age mice, they have a bit of a deficit. So when you compared young and age, the young continue learning and actually do better than they were doing before at the new location, whereas the age mice, they consistently have a deficit in the performing at this task. So this is something that we're very excited about, and is something that we're pursuing in the lab now. So basically, I'm going to talk to you. I'm going to start by talking to you about environmental aspects and how environment can affect cognition. And people have known that if environment affects cognition, but the environment is a very fuzzy term, and you need a handle to study this. And during my postdoc with Rusty, I became very interested in adult neurogenesis. And as many people here in the audience will know, there are a few well characterized areas of adult neural stem cells that produce adult neurons into adulthood. In the mouse, this would be the subventricular zone and the subgranular zone. And the subgranular zone is actually relatively well-characterized neurogenesis locus in humans too. And this area here, the subgranular zone, produces new neurons that integrate the dentate gyrus of the hippocampus. So in the subgranular zone of the dentate gyrus, there is a pool of radioglia-like cells, so these are type 1 cells, that is usually quiescence. However, every now and then, partially in response to environmental stimuli, these cells re-enter the cell cycle, and they start differentiating, proliferating, and then differentiating in a way that kind of mimics the developmental time course of differentiation, in the uterus. And this can be regulated by a variety of natural stimuli, including cognitive enrichment, running and exercise, stress, age, seizures, and so forth. Now, the question that has got the field thinking for a long time now is how does integrating new neurons actually improve cognition? So these new neurons are actually a very small fraction of all the neurons in the hippocampus. So you'd be thinking, how can you have just a few more neurons and improve cognition? And the key is probably the fact that these neurons are actually baby neurons. They're more excitable, and they have plasticity properties that are akin to the very young human neurons. And for a long time, the dominant thought in the field is that these neurons, even though they're more excitable, they actually act by surround inhibition. That is, they they engage inhibitory circuits that inhibit the neighboring cells. And in doing this, they kind of keep the activity in the dentate gyrus very sparse. So the idea was that neurogenesis increases sparsity. So these newborn neurons, they increase sparsity. And there's a variety of literature, theoretical neuroscience literature, saying that sparsity can actually be beneficial for memories. Why? Because if you have few neurons active during formation of memory, then you have less likelihood of memory interference. Because if you have a lot of neurons responding, then it's very likely that other memories will engage the same neurons. So by having sparsity, so the thought goes, you can actually improve memory discrimination. So this is what a graduate student in the lab, Agustina Verschu, set out to study. So she worked with theoretical neuroscientist at Einstein, Ruben Cohen-Kaigi, as a colleague of mine. And she decided to basically ablate neurogenesis using focal irradiation of the hippocampus in a subset of cells of mice, whereas she left another subset of mice without undergoing irradiation. She then housed these mice in two different types of housing. One is enriched environment, and the other one a regular cage, right? And then after these 2 weeks of environmental enrichment, she brought the mice back to regular cage. And then after a week and a half, she imaged the mice as they explored this treadmill. And this is the kind of recordings that we obtain in the lab. So here you're seeing the dentate gyrus, and the cells that are shaded in color, these are the active cells in the dentate gyrus during a 10-minute recording. And we get the activity of these cells, and we get information about the position of the mouse. So this is where the mouse is on the treadmill. So each one of these lines is a lap. And we see the cells the flashes here, so these peaks of calcium activity, this is the cells responding. So the first thing that Agustina checked is whether irradiation actually resulted in changes in the number of newborn neurons. Here you can see them. They're labeled with DCX double cortin, which is a marker for immature neurons. And you see that in a regular cage mouse, you get robust neurogenesis. But if you previously irradiate the mice, you do not have any neurogenesis at all. So this is very effective, despite the fact that irradiation can have other effects. And I'll talk about a little bit of this later. This is very effective at reducing adult neurogenesis. And what you see here is that regular cage animals have a certain number of adult-born neurons. Irradiated animals have none, and enriched environment animals actually more than double the number of adult-born neurons. So this is a very robust effect and has been amply described before. So now, this is what Agustina did that I think was a really cool experiment and a really cool finding. She said, okay, so now we have animals that some have increased adult neurogenesis due to enriched environment, some have decreased adult neurogenesis. How well does the environment where the animals are walking, how well is it encoded in the hippocampus? So what she did is she used a machine learning algorithm and had the machine learning algorithm associate the position of the mouse with the firing of the neurons. And then she, after the machine learning algorithm had learned, she said, okay, now I'm not going to tell you where the mouse is. You tell me where you think the mouse is. So the computer basically had to decode the position of the mouse based on the flashing of the cells. And the position of the mouse here is in the background in blue. And what you see here in orange is the estimate from the decoder. What you see is that the decoder actually does a pretty good job of figuring out where the mouse is. Sometimes it goofs, right, here and here, but most of the time it does a pretty good job of estimating where the mouse is. Now, using this very unbiased way, so it's just a computer trying to do the estimate, what we found was really surprising. Is that enriched environment mice actually the computer is able to decode activity much better in these mice than in regular cage mice, and much better than in irradiated mice. So using this very unbiased method, you can actually, our decoder can actually extract a lot more information from the hippocampus of these animals that went into a rich environment. And this is telling us that these animals actually encode more spatial information. So we looked at this at a single cell level, and we plotted all the single cell Fisher information, which is a measure of a lower bound estimate of the information content that each cell encodes. And what we saw is pretty much the same thing. So regular cage and enriched environment mice have a lot more information, carry a lot more information than the irradiated cohorts. And this we found is across the whole range of information content. So it's not only like the place cells that are very tuned to a single location that they carry more information. It's all of them. Even the cells that are not very well tuned to a single location, they carry more information about location than in the irradiated cohorts. And what was really surprising, based on what I told you earlier, was that activity was actually highest in the animals that had a lot of neurogenesis, enriched environment animals and regular cage animals. So this got us thinking, it was very puzzling. Why? Because I told you at the beginning that the way that the field looks at this is by this idea that adult-born neurons are actually silencing the hippocampus and contributing to keep activity low. But here we're actually seeing that the activity of individual neurons is actually higher on average. So we decided to go and look at the tuning of cells and see what was going on. So you have, in any recording, you have cells that are not very tuned and cells that are highly tuned. This is a plot of the whole recording. This is about 10 minutes of recording, and what you're seeing here is the activity of these neurons. And it's plotted. The circle represents a full lap. So this cell, for example, over all the laps was very consistently active as a single location. This cell was randomly active everywhere, right? So this is a tuned cell, this is a non-tuned cell. And Agustina went on to fit these tuning curves, which is what they're called, with von Mises functions, which is basically a Gaussian in polar space, right? And what she found really completely reflected her previous findings. So enriched environment neurons had the highest fit to a single Gaussian, a unimodal fit, followed by regular cage animals, and then irradiated animals had the lowest. So the next thing that Agustina asks is, what does this mean? Does this mean that the firing of these place cells is maybe not as consistent? So if you think about, the animals could fire maybe not in the same spot consistently, but maybe sometimes a little bit ahead, sometimes a little bit behind. Or they could be firing very consistently at the same spot. And this is manifested in the width of the tuning curves. But she found no differences in the width of the tuning curves, meaning that these cells are firing consistently at the same spot. But what she found is that when they do fire, they have higher transients. So the amplitude of the tuning curves is higher. And this kind of changed how we look at adult neurogenesis, because we now think that newborn neurons are actually modulating the gain of the dentate gyrus. So if you think about it, it's a little bit like the volume dial. You're actually increasing the volume like an amplifier. And in this way, in cells that are tuned, you are increasing the signal-to-noise ratio. So Agustina then went on to, because irradiation has a lot of side effects, she went on to develop a system to silence these neurons with chemogenetics, with HM4D, which is basically a derivative of a human muscarinic receptor that can inactivate neurons. So she expressed, using a genetic approach, an ASCL1 mouse. She was able to target this receptor, this engineered receptor, to adult-born neurons. She tested it using contextual fear conditioning. In contextual fear conditioning, you have the mouse associate a foot shock to a particular location, and then you expose it to another location, a similar but different location, which is this other cage with the little hut inside, and you measure whether they freeze or not. Freezing is the mouse's natural response to fear. And if the mice are very fearful because they think, wow, this is the place where I got that foot shock, I better just freeze, then they'll freeze. If they say, well, this looks a little bit like the place where I got the foot shock, but I know it's not the same, then they won't freeze, or they will freeze less. And this is exactly what we found, that with usually mice will freeze in the context where they got foot shock, but not the different contexts. But if you silence adult neurogenesis, this discrimination index is produced. So they're not able to discriminate between where they got the shock and where they didn't. And Agostina repeated all of these experiments, both in single cell fissure information, activity, goodness of fit, and width of distribution and amplitude. And she really found the same results. So by inhibiting adult-born neurons, she actually found lower activity, reduced amplitude of tuning curves, and lower spatial information. So I'll just go over this very quickly. So our findings is really that environmental enrichment increases the spatial information in the dentate gyrus, and we showed that this is really due to adult neurogenesis. This effect is mediated by adult neurogenesis, where we find something specific to ascribe this effect of environmental enrichment to. And we found that ablating adult neurogenesis actually decreases single-cell spatial information, and it decreases the activity out of individual cells. So we postulate that newborn neurons actually act as gain modulators of the dentate gyrus. So now we're kind of in a position to replace this former model with a more nuanced model, where you probably have some sort of excitatory feedback going back into the dentate gyrus that is allowing this gain modulation and actually increasing the activity of neurons when the adult neurogenesis is present. But how does this actually work? It's not fully clear, but we think that it's probably mediated by mossy cells, which is something that we're looking into right now. So moving on, the question we asked next is, what's happening in aged animals? So aged animals do not have, they basically have no adult neurogenesis, because adult neurogenesis decays a lot during aging. So this is work done by Kelsey McDermott, who's actually here in San Diego now, working at SBP. And Kelsey basically found what many other people have found, that aged animals actually have impaired spatial memory. So we use a lot of this spatial memory test, which is called a displaced object or displaced object test, where you train the animals to explore a box with two objects, and then you displace one of them, and naturally the mice are curious, and the next time they go there, they will tend to spend more time exploring the object that moved, right? And what we found is that this is gone in aged animals, and this is compatible with a lot of findings that people that in the field from aged animals. So what Kelsey did was she recorded the first, what we think are the first recordings from aged animals. So these are animals over 20 months old, from the dentate gyrus of aged animals. And this is quite amazing that she was able to do this, because some of the animals were well above 20 months, and we were afraid that the animals wouldn't survive the implant surgery. But they did. And what she found was that activity in the dentate gyrus was increased in the aged animals. And this is actually compatible with a lot of previous work on activity in the aged brain. And it's this idea that the aged brain is hyperactive, probably because of dysfunction of modulatory circuits and inhibitory circuits. But what she also found is that the aged brain has a sharp decrease in spatial information, meaning these animals don't encode space, the treadmill, as well as the young animals. What was really interesting, though, is this bit of a nuance, that the animals initially do not encode the treadmill very well. But as you start running the mice on that same treadmill belt over time, they eventually pick up, and they will, after 4 days, they're pretty much undistinguishable with the young animals. So it's not, the way, this is not really learning, but the way that we try to explain this is saying, well, it's not like age mice can't do it. They just need more time to do it. They're eventually, they will form a representation of the treadmill over time. But what's really interesting is that even though people have in the field, there is this idea that the hyperactivity could be a cause for a lot of the cognitive deficits. What we found is that the hyperactivity doesn't actually correlate with the encoding of the treadmill, meaning you can have the hyperactivity that is typical of old age, but not have deficits, because after 4 days, they do have good encoding of space while still manifesting hyperactivity. We also looked at reactivation rates. So this is basically the idea that once you expose the animal to a treadmill, a certain subset of cells will be active, and then the following day, there's a higher likelihood that these cells will also be active again, and the day after that also. And we actually didn't see any changes in the reactivation rates. So the reactivation rates were exactly the same. So basically, from Kelsey's work, what was really interesting is this idea that spatial encoding can actually be good in aged mice. So whereas young mice get the spatial encoding at a certain level immediately from the first session, after 4 days, the aged mice catch up. And that they do this even in the absence of, even with the deficits in terms of excitability, which is really surprising to us. So one of the possible causes for these deficits in aging is inflammation. Systemic inflammation goes up during aging. And Kelsey was also very interested in looking at, what is happening to the encoding of memories in the hippocampus, the encoding of space in the hippocampus, if we have a systemic inflammation. And this led us to ask the question, microglia are involved. Microglia are the resident macrophages of the brain, and they're involved in a variety of immune responses, but also they're involved in homeostatic maintenance, which is something that has come to prominence in the last 15 years or so. So we now know that microglia are actually involved in pruning synapses. Synapses are no longer used, and they actually can stimulate the formation of new synapses and the removal of synapses. However, the molecular pathways that mediate this response are common with the immune responses. So there could be some crosstalk. This creates a problem. There could be some crosstalk, meaning that if you have a strong immune response in the body because you have generalized inflammation, you could actually be damaging synapses. And this is the question that Kelsey tried to answer, do you have changes in the encoding of information in the hippocampus if you have systemic inflammation? So in order to study this, Kelsey injected LPS, which is a surface molecule derived from E. coli bacteria, and this produces a very strong inflammatory response that is systemic. She injected mice with LPS, and the LPS will, in the immune cells and other cell types, will will activate TLR4 receptors and induce the secretion of inflammatory cytokines that will go everywhere, including the brain, especially IL-1, IL-6, TNF-alpha. And we don't think that LPS directly crosses into the brain, although it can sometimes if the immune response is strong enough to disrupt the blood-brain barrier. LPS is known to alter microglia morphology. So basically, microglia morphology is a good indicator of the activation state. So homeostatic microglia are usually very branched, and they are surveilling the environment around them. However, after LPS injection, they become more amoeboid. So they have smaller arborization. And as you can see here from this shoal plot of their structures, of their morphology, of their branching, and you see that with only 2 LPS injections, 2 days of LPS injections, there is a significant change in the peak of their shoal plot, and the total number of interactions, and the total branch length of their processes. So these are all hallmarks of immune activation. And what this leads to also is a decrease in the number of synaptic spines in the hippocampus. And here we're looking in a hippocampal area CA1. So, okay, we know that these injection with LPS will trigger this response. Now, the question is, what does this mean for the encoding of information? And this is what Kelsey looked at. And what she found is some modest changes in activity levels, which are a little bit increased, but a strong decrease in single-cell Fisher information. So meaning spatial information is highly reduced in these animals. So we know that this synaptic remodeling, the removal, the pruning of spines is dependent on the complement cascade, complement signaling. And in particular, synapses are due for removal are tagged with with complement 1. So what Kelsey did was ablate complement C1q, which is expressed and primarily in microglia in the brain. And she ablated C1q specifically in microglia by crossing C1q flox mice with CX3CR1 mice. Now, this approach will also target peripheral macrophages. However, those get replenished. So there's an inducible system that is inducible with tamoxifen injections, so the recombinant peripheral macrophages will eventually flush out, leaving the microglia, because they're resident in the brain, as being the ones that actually lack C1q. So she, after selectively ablating C1q in microglia, what she found is that there was an increase in activity levels, and again, a sharp decrease in single-cell Fisher information. That is, these animals don't encode spatial information as well in the hippocampus. She went back to our old friend, our behavioral assay, spatial behavioral assay, and she found that these mice had spatial behavior deficits. So if you ablate C1q in microglia, you will actually you're not actually able to form spatial memories in the same way as with intact C1q. So she also looked at the morphology of microglia in these ablated mice, and what she found is that they have they basically have a more branch phenotype. That, I just told you that less branch phenotype reflects less more inflammation, and more branch reflects more inflammation. So she found differences in terms of in the peak of the shoal plot, and in terms of the number of intersections, the total branching, and even in soma volume. And she found both in CA1 and in the GG, she found an increase in synaptic density, which is compatible. So if C1q tags synapses for removal, and C1q is ablated in microglia, then you expect less removal of synapses, which is exactly what we see, that it would result in an increase in synaptic density. And furthermore, it results in an increase in synaptic material. So here we're labeling microglia, we're labeling brain tissue with Ph.D. 95, and we see more Ph.D. 95. Ph.D. 95 is a postsynaptic density 95 is a marker, a synaptic marker, and we see less of these synaptic markers in microglia that express C1q. The question that Kelsey asked next was, well, if you increase inflammation, you have less you have less spatial information. You decrease inflammation by ablating the C1q, or the inflammatory response by ablating the C1q pathway. What happens is that you have less information encoding. What happens if you do both? So what she did is that she took these conditional knockouts for C1q, and she injected LPS. And what we found is that is actually somewhat surprising, because the LPS injection and the C1q conditional knockouts actually normalized synaptic density. So basically, the knockouts are immune to LPS. So if you have if you ablate C1q for microglia, you normalize the spine phenotype. And we didn't find significant differences in terms of single cell activity, but we do see that single cell fissure information is normalized. So in this model, basically what we're seeing is that if you have too much inflammation, or if you block the inflammatory pathway, you reduce spatial information. However, the inflammatory response seems to be driven uniquely by C1q in microglia. So despite redundancy from astrocytes from other cells that can mediate the inflammatory response, we can correct it just by removing C1q from microglia. This means that basically C1q is a potential therapeutic target, modulating C1q in microglia, modulating the microglia immune response, despite other cells that are also involved in the immune response in the brain. So this is the gist of what I just told you, is that spatial information encoding and spine density are impaired in a model of systemic inflammation, but also in animals with selective ablation of C1q in microglia. And if you ablate C1q in microglia, you counter the effects of inflammation on spines and hippocampus information encoding. So this work kind of highlights that there's this balance of pro and anti-inflammatory signals that modulate memory formation circuits in the hippocampus, but what is particularly important is that they do involve microglia. So despite a redundancy, some redundancy that has been described in the literature between microglia and other cells, we are able to completely restore spatial encoding by only affecting C1q in microglia. So as we started working with microglia, we actually got very intrigued on the ability of microglia to actually be engineered to address cognitive deficits. So if microglia can have such a powerful effect on the encoding of information, can we actually engineer microglia to address cognitive deficits and disease? And this is an area that is extremely popular right now. There's a lot. This paper is from 2025, and this group, they're based in China. They actually did microglia replacement in humans by doing bone marrow transplantation. So ablation of microglia using a CSF1 receptor, inhibitor, and repopulation after bone marrow transplantations. And this was done in a cohort of patients that have a rare but lethal disease that involves that is caused by mutations in the CSF1 receptor. This is really a remarkable thing, that you can actually remove microglia. You can completely remove it by using a CSF1 receptor antagonist and repopulate with exogenous microglia. So we thought, maybe we can explore this by engineering microglia to deliver in a controlled manner, deliver a neurotropic factor in the adult brain. And this is actually a paper that is currently under review. It was work led by Rowan Offlin in collaboration with my colleague Jean Hebert, who's now no longer at Einstein. He's gone on to ARPA-H. But the idea was that we could completely remove microglia in the adult brain, transplant new microglia that express BDNF, which they don't usually express, and look at the effects of BDNF in the brain. So you can, as I said, completely remove endogenous microglia, and you can do this, and you need to do this in order to transplant microglia. So if you transplant microglia in mice that have, or animals that have endogenous microglia, the contact inhibition of microglia will prevent the transplant from spreading. However, you can actually completely remove microglia with, or nearly completely remove microglia, either a PLX treatment. This is usually PLX 5622 or 9733. These are just drug names, but they are CSF1 receptor inhibitors, and you can kill off all the microglia. But there's always a few that survive, and even these can be really a hindrance to transplantations. You can also do a genetic deletion of microglia by basically removing CSF1 receptor specifically in microglia, and this will give you an almost complete removal of microglia. So what we did is using this genetic approach, we started, we as a proof of principle, we looked at what happens when you transplant these new microglia into the brain. So we took donor mice that have green microglia that express GFP. We cultured them, and then we injected them into a adult host mouse, and then examined the brain after 14 days or 1 month. And you see that at 14 days, you see this wave of propagating microglia that is repopulating the brain. You see here how the density is higher. And by 30 days, most of the cortex and hippocampus have been completely repopulated by the transplanted microglia. These transplanted microglia are a little bit different from the resident microglia that the mice are born with. So they actually mostly resemble repopulating microglia. And the repopulating microglia are the microglia that repopulate the brain once you ablate microglia. So they're thought to come primarily from surviving microglia in the brain and from peripheral macrophages that infiltrate the brain when microglia are not present. And what we find is that in terms of morphology, basically, these are the endogenous, the microglia the mice are born with, and you see that their morphology is mostly resembling the repopulating microglia, which are these RMGs. And the transplanted ones mostly resemble these microglia. Also, in terms of transcriptome, we did a lot of single-cell RNA-seq, and we see that these transplanted microglia, they actually cluster with the repopulated microglia, but they're different from the resident endogenous microglia of the brain. And several groups have found the same thing. However, I'm told that maybe if we had waited for over 1 month, these cells that we transplanted, they would indeed resemble the endogenous cells. And there is some conversation in the field about this, with some groups saying that if you look later on, that the transplanted microglia can actually completely resemble the endogenous microglia that the animals are born with. I don't know the answer to this yet, but this is something that, I don't think these papers are out, but this is something that people are discussing in the field. So one of the problems that for applying this technique of transplantation and microglia replacement, one of the problems that I mentioned is that whenever you do this with a drug, and there's actually FDA-approved CSF1 receptor inhibitors on the market, PLX 9733 is one of them, and you actually need to do this treatment with the drug to condition the brain by ablating microglia completely. And what we found is that, I told you that a single round of this treatment is not enough. What we found is that if you do pulsatile treatment, so you basically put the mice 1 week on the CSF1 receptor inhibitor, 1 week off, then 1 week again, then 1 week off, then 1 week on, and then in the end you have effective removal of microglia, and you have good conditioning of the brain for transplantation. And there's actually, so we see that after 3 rounds, and funnily enough, if you continue doing this, you don't get as good transplants, and I don't know why. I cannot explain why more pulses is not as good. The sweet spot does seem to be 5 courses of treatment, and it's really interesting because, as I told you, this field is very, very moving very, very fast. And we had a bunch of these, this paper is under review, but we had a bunch of other papers actually that kind of had similar findings to us in the meantime. And there was this paper that appeared last summer, and they found exactly the same thing using a different CSF1 receptor inhibitor, but they found that the three pulses of PLX inhibition is actually the best for repopulating the brain, and it achieves almost complete removal of complete ablation of microglia, of the endogenous microglia. So this is one way of being able to improving the transplantation of these cells. The second innovation that we tried was using a constitutively active CSF1 receptor that can be transiently induced, we can induce expression in the repopulating cells. So we infected these transplanted microglia with a lentivirus encoding for this constitutively active mutant. And what we found is that if we induce the expression with doxycycline, these cells that have the receptor, actually they do better. They have a competitive advantage, and their area spread is higher than if we don't do the induction of the constitutively active receptor. So we found a slightly higher cell density and a much higher spread, and they seem to spread faster. We were hoping that the constitutively active receptor would provide resistance to PLX, to the inhibitor. However, this wasn't the case. So the cells still do not spread as well, even if they express this constitutively active CSF1 receptor. They don't spread. So, because we were hoping that we'll be able to mitigate the competition between endogenous cells and the transplanted cells, but this didn't happen. But we still got an advantage. And so using this, so what Rohan has shown is that he can actually deliver BDNF to the brain in microglia in an inducible fashion, where he induces with doxycycline, and he increases tissue BDNF levels. And most importantly, if he looks at the tissue, and I know that I'm running out of time, but most importantly, he increases, so tract B is the target of BDNF, and he increases phosphorylation of tract B in the whole brain tissue, cortical tissue. So as a summary, the transplanted microglia are transcriptionally, morphologically, and functionally similar to repopulated microglia-like cells, which other labs have also shown. So we have the same things after 30 days at least. Question is still whether, if we keep the transplants there for longer, whether they'll approximate the endogenous microglia. 3 pulses of CSF1 receptor inhibition can ablate microglia and condition the brain for repopulation. And this really seems to be the sweet spot, at least in mice. And the transplanted microglia expressing a constitutively active CSF1 receptor mutant have a competitive advantage and show enhanced repopulation. And microglia replacement can be used for the controlled delivery of BDNF in the adult mouse brain, which I think is a first of being able to deliver a large peptide in a controllable fashion. And we've shown also that you can actually, this can lead to increased phosphorylation of tract B receptor. So this is a functionally active transplant. So I know I'm running out of time, so I just want to take a moment to acknowledge the people who did this work. Agustina Fresh, who did most of the neurogenesis work that I described. Kessie McDermott did aging and Siwan Key's story. Jake Jordan did a lot. He's now, so Agustina is at Rockefeller. Kelsey is here, SBP. Jake is at Eli Lilly, and he did a lot of the setting up of our behavioral setup. And Rowan Offland led the story with the microglia transplantations. Also thanking my collaborators and our funding sources, and thank you for listening.
Very nice talk. So I had a couple of questions. One was about the microglia, and basically with the microglia resident in the brain, they can secrete chemokines. When you transplant and when you treat with that CSF1 receptor antagonist, it'll wipe out peripheral blood monocytes and myeloid progenitors actually as well. So I'm wondering if you actually have more infiltration of monocytes from the periphery that are contributing to the microglia pool, because we're doing we've done a clinical trial with Stephanie Cherky where she gives hematopoietic stem cells that are modified with a gene to replace what's missing in hereditary cystinosis. And those stem cells track to the brain and they actually have an effect. So when you showed your dendritic spine photomicrograph, it looked like there were more cells in there, more GFP positive cells.
So the GFP positive, so for those spines, those are neurons, and those are
Yes.
neurons that are actually infected with a lentivirus. So there could be different density depending on the area. So you actually see a little bit more background.
Yeah, that's what I was wondering about, because
Yeah.
so I'm just wondering, is there more neurogenesis going on?
So these are not newborn neurons. This is in CA1.
Okay, got it.
So they're mature neurons, and the reason why they're, when I was putting that together yesterday, because this is a new slide that I added, and actually it's a technicality. So
Okay.
it's not it's just because that area had higher density.
Got you. I had another question, and that's about fear learning. So when you show that the mice don't really know where they got zapped, there's a new paper in Nature yesterday. I know it's not fair because it just came out yesterday.
I did not read it.
On astrocytes and the amygdala and how they affect neuronal responses. Have you looked at non-neuronal, in other words, astrocytic responses?
So we haven't actually done any, but we're doing some now. We're using more of contextual fear conditioning in our experiments. We only initially used it for the Neurogenesis project. For the other projects, we mostly did the novel object and object location, memory location. So, but we're planning to. I mean, I think it's interesting that they see that people are seeing all of these deficits. And I think it's very interesting, we're talking about the context, even in the context of cancer and what the cancer does to the brain and generalized inflammation. So it'd be a lot more interesting to study this. What was really surprising, though, was that just by eliminating C1q from microglia alone, you actually no longer see this effect of LPS in the brain. Because there are other immune cells that could be mediating that too. And we know that there is some redundancy, and if microglia are not active, then astrocytes can take over for pruning.
Let's do an online one. Here the questioner wants to know, do we know the extent of adult neurogenesis and spatial information processing in immune deficient mice, for example, the RAG knockout?
No, that's a great question. And actually, we're trying to bring this. We're trying to bring this microglia transplantation work to humanized mice, and that will be something we'll be looking at. So basically, neurogenesis is known to be dependent on fractal kine signaling. So there are differences in microglia. There's some literature. Amanda Sierra, who's now in Spain in the Basque Country, has some beautiful papers about how neurogenesis is actually influenced by microglia and by inflammation. And there's a big decrease in adult neurogenesis when there's neuroinflammation. Yeah.
Fantastic talk. I had a question. So as I understood, new neurogenesis specifically is related, the excitability of these new neurons is a crosstalk between the inhibition of the dentate gyrus. Does the dentate gyrus inhibition go up over life in that it's not that you see less neurogenesis, but that you see more inhibition from the dentate gyrus?
No, actually, I think that what people see is that less inhibition in general, and that's what we also showed in our aging, where there's a hyperexcitability. So we think that this is due to reduced inhibition, and I think that this is common across a few brain areas that inhibitory circuits are less active in aging, and therefore you actually have higher propensity for seizures and so forth. Yeah.
Sorry, I had a couple questions. One is about the C1q. That's an inducible system, right?
It's an inducible
So
system.
if you looked at is there a developmental system where there's a knockout of C1q in mice?
There's a developmental knockout. You can use the flocks
And will that
This is in
developmental
use.
knockout, you think, have the same phenotype, or the fact that it's knocked out since birth?
This is controversial, right? So, there's all these papers going back 15 years or so. Doris Schaefer and blanking on her name at Harvard. Anyway, all these papers looking at the pruning of spines by microglia, right? Of synapses by microglia. And it's supposedly really important during development, right? Because during adolescence in humans and in mice, you have this massive pruning event that reduces the number of synaptic connections. And now there is these fire mice that lack microglia from birth, from ever, so they never actually have microglia. And it turns out that the mice are supposedly doing fine. So there's a group from Edinburgh that published this maybe a year ago or so. I don't know exactly what the latest is, but the story seems to be a little bit more complex, and why exactly, whether it's a technicality, whether it's something trivial, but there is a discussion about what happens if you never have microglia developmentally, because you would expect a big deficit. However, this group says that there isn't. So
I meant specifically to your results, and for example, the LPS result. Do you think it matters if the C1q is knocked out? You're born without C1q, or you do it later in adulthood, as you did?
No, I think that with the LPS, I think just that I think what you can say is that I would expect the animals to be cognitively impaired if they lack C1q and microglia. Because that's what we see, with the memory. So you have no C1q and microglia, and you have an impairment. I think that would be true also developmentally, if it's development from onset.
There's a clinically relevant question. The questioner wants to know, he or she understands that you're focusing a lot on the hippocampus in terms of spatial learning and all of that. Clinically, the parietal lobe plays an enormous role clinically in spatial orientation of patients. And do you have any speculation? Does the hippocampus crosstalk with parietal lobe?
Oh, yeah.
Is the exact same thing happening?
Sure. I mean, so one thing that is important about this, everything that I told you, is that there's important differences in microglia depending on the brain area. We're doing these knockouts, these conditional knockouts all over the brain. So we're basically doing it's everywhere, and we're not taking that into consideration. So some of the effects that we see could actually be mediating by things that are happening elsewhere. But yeah, so yes, spatial orientation, there's other areas that are involved. One thing that I don't think I mentioned is that, this is something that we started talking about, collaborating with some of our neurosurgeon colleagues, because people do these place cell recordings, and we're doing this at Einstein, place cell recordings in humans. So the idea is then you'd be able to get tissue from these surgeries, and also have the cognitive tests and the place cell recording. So would be a very exciting thing to do.
All right, it's been a great discussion. I think we better close it right now, and thank Tiago for a really stimulating talk.